Good evening to all listeners and viewers. Myself, Dr. Ichapura. Today, I want to share some information about steps of DNA replication through the PowerPoint presentation. DNA replication is a fundamental process by which the parent DNA duplex copies itself and become duplicated to produce two daughter DNA duplexes. DNA replication takes place during synthetic phase of interphase so that the two daughter cells after cell division get equal amount of DNA as that of parent cell. Of the various ideas proposed regarding mode of replication, the semi-conservative mode is highly accepted. It has been theoretically predicted by Watson and Crick 1953 on the basis of double helical nature and complementary sequence of bases on two strands of DNA. This slide shows the three modes of replication. The second one, semi-conservative mode, is highly accepted. For it, the two strands of the parent duplex separate and act as template on which the new strands are assembled. Semi-conservative mode of replication has been later experimentally proved by Michelson and Stahl 1958 using N15 in the nutrient media for culturing Escherichia coli and monitoring the density of DNA after each replication. For semi-conservative mode of replication, the two strands must get separated by dissolving hydrogen bonds between base pairs gradually in a zipper-like manner. Then two separated strands act as templates for synthesis of new strand by selecting the complementary deoxyribonucleotides from the pool present in nucleoid or nucleus. Of the two strands, one is parental and other is daughter. Replication starts at origin of replication, appear as bubble under electron microscope, ultimately extend in the form of two Y-shaped replication fork. Replication takes place bidirectionally from the origin of replication. Now this slide shows that in prokaryotes, as the DNA is circular, there is only one origin of replication, whereas in eukaryotes DNA is linear and quite large and has multiple origins of replication. In both, replication takes place bidirectionally. Now proteins and enzymes involved in replication. The enzymes and proteins are DNA gyrase, which helps helicase in unwinding. Topoisomerase creates nick, prevents supercoil formation. Helicase locally opens double helix by breaking hydrogen bonds. Single-stranded binding protein stabilize the single-stranded structure. RNA primase synthesize RNA primer. DNA polymerase 3 synthesize daughter strand in 5' to 3' direction. It is helped by sliding clamp to move ahead. RNase dismantles the primers. DP1 fills the gap with complementary deoxyribonucleotides and DNA ligase joins the two DNA stretches. Mechanism of replication was studied for the first time in Escherichia coli. Replication in higher organisms is less well understood but involves almost same type of mechanism with some differences only. For initiation of DNA replication, recognition of origin of replication is done by pre-priming complex consisting of helicase and single-stranded DNA binding proteins. Now let us see the steps. For replication to take place, the two strands of DNA double helix at weak spots where there is more adenine-thymine base pairing separate, dissolving hydrogen bonds by the enzyme helicase. The opening is facilitated by topoisomerase, which creates a nick or cut in one of the two strands by breaking one phosphodiester bond near replication fork. The nick prevents supercoil formation in DNA double helix on separation of the strands. This is the nick, and the diagrams show helicase, single-stranded binding proteins, topoisomerase. The point where the DNA is separated into single strands and where new DNA will be synthesized is known as replication fork. Next step, the separated strand localized opening appear as bubble under electron microscope having two Y-shaped replication fork with neck and mouth. In prokaryote there is single origin, whereas in eukaryotes there are multiple origins. Now this is the origin magnified, and in the bubble shows two Y-shaped replication fork. The single-stranded DNA structure of replication fork is stabilized by single-stranded DNA binding proteins which act in cooperative way. DNA polymerase 3 starts synthesis by adding complementary deoxyribonucleotides onto 3-OH end of RNA primer. For this, a short sequence of ribonucleotides, RNA primer of about 10 nucleotides is synthesized prior to DNA synthesis by primase, having a free 3-OH group which acts as first acceptor of incoming deoxyribonucleotide introduced by DNA polymerase. Primer is required because DP is unable to initiate the formation of DNA strand de novo. Rather, it is capable of adding nucleotides to a 3-OH terminus of the existing strand provided by RNA primer. As DNA polymerase can only add deoxyribonucleotides in 5' to 3' direction, and as two strands of DNA double helix run anti-parallel, so synthesis on two strands differ. On one of the strand of replication fork running in 3' to 5' direction towards fork, DNA is synthesized continuously in 5' to 3' direction using only one RNA primer with its free 3-OH end at the beginning by DP3 movement along the template. It is called the leading strand. On the other strand of replication fork running in 5' to 3' direction away from the replication fork, DNA is synthesized in a discontinuous manner in 5' to 3' direction away from the fork as small stretches. Each time it requires RNA primer with its 3-OH for adding deoxyribonucleotides by movement of DP3 along the template. As a result, many RNA DNA pieces are formed which are termed as Okazaki fragments after the discoverer Okazaki, Japanese molecular biologist. This strand is termed as lagging strand. Although both strands replicate simultaneously, but DNA synthesis on lagging strand require help of another two enzymes. RNase dismantles first the primers and then DNA polymerase 1 replaces deoxyribonucleotides to fill the gaps. After that, DNA ligase catalyzes phosphodiester bond formation between two successive DNA pieces, as a result of which continuous DNA strand is synthesized and thus replication on lagging strand is completed. As both strands synthesize DNA simultaneously, it is presumed that two molecules of DP3 move in opposite direction along their respective template strands. Okazaki fragments have RNA primers and DNA stretches. For ligation, first RNA primers are removed by RNase and the gaps are filled by deoxyribonucleotides by DP1. DNA ligase establishes phosphodiester bond between two DNA pieces. As a result, continuous stretch of new DNA is synthesized on lagging strand away from the replication fork. In this way, replication is completed on both the strands replicant simultaneously and two daughter DNA duplexes are resulted. In eukaryotes, replication starts at multiple origins simultaneously, proceed bidirectionally and ultimately meet with each other. In this way, entire linear DNA is replicated. Now this slide shows the replication fork. This is the leading strand having only one RNA primer, and this is the lagging strand having many RNA primers, and on each RNA primer DNA is synthesized and RNA DNA bits are known as Okazaki fragments. From these Okazaki fragments, later on RNA primers are removed by RNAs and gap is filled by DP1, and then the pieces are sewed with each other by means of DNA ligase. Now this diagram shows the summary of DNA replication, the enzymes involved, and let us see it with the help of diagram on whiteboard. This also shows that on leading strand the DNA is synthesized continuously and only one RNA primer is needed. On lagging strand, synthesis is discontinuous. Many Okazaki fragments are formed, each having RNA primer at the 5' end. From these RNA primers are removed and gap is filled by DP1, and then the DNA stretches are sewed by DNA ligase. Now, in this way, the steps of replication have been summarized.